So going through number one, that's a diagram that we had with the positive at the bottom, negative at the top. And you can see each one. So this has 3, this has 3, this has 4, and the last one has 3. For B, it'd be the denaturing step of the PCR would denature the bacteria and would not synthesize the DNA. For C, it is DNA helicase would have no effect because the heat is what separates the DNA strands. And then for D, termination would only take place if the T base pairs in the template strand, and only the DD ATP would be seen. So to fill in the blanks, the first one is DNA synthesis, then the primary annealing step. For B, it is the AMP-R and restriction enzymes. C is the positive electrode. For D, it is the 3' and then the 2'. E is transformation. F is the IPTG, and then the X scale. For 3, so you can see, I'm not going to read off every single one, but here's the top, and then there's the bottom. And then the top for B, top and bottom again, there's the calculations, so it would be 59.81 and 62.375. C, again, basically I could just say there's 3 strands for the top and then 6 for the bottom. And then D, those 2 proteins, really wouldn't be included because the heat is what's used instead of those. For 4, again, here's the diagram, I guess. So 3 in the first, then 2, 2, 4, 4, and 3. And then here's the band, and we have 50, 150, 100, 100, and 500 with band HI, the echo RI on the hint right there, and the band again. And then the fragment lengths would be 50, 100, 150, and then 500 base pairs. And then for number 5, so these are the steps. So basically, I'll just go through real quick. So you'd purify a mature mRNA from a sample of cells, and then you mix it with the primer. Then you'd add reverse transcriptase and the DNTPs to synthesize a strand of DNA. And then you add RNase H to digest the mRNA, and then you add the DNA polymerase 1 to synthesize a second strand. And then to clone, you'd have a plasmid and chromosomal DNA with the YFG gene that are digested with the same restriction gene. Then the plasmid and DNA molecules are mixed, and the DNA ligase is added. Cells of E. coli are transformed, and then the growth occurs on a medium with ampicillin. And then the last one, growth of bacteria with a medium that has ampicillin and IPTG. Then you can identify the colonies that have beta globin insert. And then for number 6, this is kind of the research I did. It was just kind of a summary, but so there it is. I put this new technique stands for clustered, regularly interspaced, short palindromic repeats, and then the CRISPR associated protein number 9. This new technique is in improved form because it's faster, more accurate, and has proven to be cheaper. This technique captures small segments of DNA from viruses and allows the bacteria to remember the viruses. And if they attack again, then the bacteria produce RNA segments and send them to attack the virus's DNA. And then the researchers are using it to make custom DNA sequences by replacing segments and making changes to pieces of genetic material.